terms of so you're going to be chairing this thing. You're going to be working with who's going to be your question people? Just Michael True. Okay. He's got the mics. So for that, if you need to go to volume for the lectern mic, it's over here. If there's feedback, with the radio mic, you can just turn those off. That's all there is to know. So you're just going to switch to the JLab now. Once I'm done.
And if Sheng does want to use the
if he wants to use that, it's already it's already wired to go. So you just have to turn it on, and it should already be on the right frequency. Just think here.
So switch back to
Yeah. It's hard to know which side this is. Are you going to switch on the computer?
Yeah, just one.
Yeah.
That's fine.
Some people like to switch.
Oh, yeah.
Okay, good morning. So it's a beautiful winter Queensland morning. Happy to see the sun is out. It's supposed to stay like this, I think, for a couple days. It'll be sunny. Maybe I think the wind's going to rise a bit tomorrow. It'll feel a bit colder, but I think it'll stay sunny. So no more rain for now. So we have a great schedule today. So the first will be a keynote lecture by Sheng. I'm just going to run through just very briefly the schedule just to put you in business here. So then Sheng, then there's going to be a coffee at the Global Change Atrium. I'll show you a map very briefly about where that is. Then a lunch. Then the concurrent sessions, which is in a different area. I'll show you a map, a lunch. Then the afternoon concurrent session. I'd like to point out the symposium in the afternoon on consciousness across species back in this room in the AEB. A pizza dinner, again, at 6 this evening, over here as well. And a special talk this evening, Songlines of the Western Desert. I hope you attend that. So this is a pretty unique opportunity. And then finally ending the day with the student social at the Regatta Hotel, which is a couple stops down by CityCat on the river. This is there'll be dinner, there'll be drinks provided. So I encourage you to go to all of that. Here's a map. Now you know where you are, the Advanced Engineering Building, there it is. There's the Lakeside Terrace. But what I need to point out is where the concurrent sessions will happen is in another building. It's called the Steel Building. You basically have to walk across that lawn, walk up those Mayan stairs, and just to the right, you'll see the Steel Building, and to the left will be a bookshop and cafe. It's pretty straightforward, I think. There's a building then there called the Global Change Building, and that's where the morning coffee will be. So the morning coffee, there'll be pastries and coffee and tea, kind of to attract you there after this morning session here for the keynote. So the coffee will be there, and immediately after the coffee in the Global Change lobby, atrium, then you'll be able to go to the concurrent session talks, which are in the colored rooms there according to what's on the program. I think it'll be pretty straightforward, and they're well indicated over there, but you'll have QBI volunteer guides to show you around as well. These are the concurrent session rooms over there. As you can see, the Global Change Institute, which is where the morning coffee will be, and then the larger A-stream session room 206 on the ground floor, then the smaller on the upper floor B-stream session in blue, 309, and then the C-stream session, 329, also on the upper floor with indicated walking paths to get between them. Again, you'll have volunteers to help you out, and there'll be signs there to show you where to go. Lunchtime is open for people in terms of going to university venues. There's pizza cafes, there's different eateries, there's the student union area. If you want to have a sit-down kind of sensible lunch. But for people who just want to grab something quickly for free, there'll be a barbecue on the lawn here, or what's called in Australia a sausage sizzle. So if you just want to get something quickly and not pay for it, Luke Sartor, one of our psychology, or one of our students here, will be just doing a sausage sizzle. It's kind of a strange Australian tradition. They take a sausage and fold it in a piece of bread and put onions on it. And in the beginning, I was just shocked at the primitiveness of it, but actually it's quite tasty. All right. So Abby Nadam will be introducing the keynote speaker.
All right, good morning, everyone. Hope you're ready for the big day that we have packed in this morning. So to kick things off, I'll be introducing our keynote speaker, which is Professor Sheng. After studying biophysics at the University of Science and Technology in China, Sheng moved to the University of California in San Diego. Here he completed his PhD in studying the early visual system. From here, he moved to Harvard as a postdoctoral fellow to continue his studies in this area of visual awareness, visual attention, and the neural mechanisms of vision. Currently, he heads the Vision and Attention Lab at the University of Minnesota and is also a professor of psychology there. Here, he has published many works in top journals such as Journal of Vision, Current Biology, Nature, and many more. And his current work focuses on the sensitivity and control of the visual system, visual attention, the neural basis of visual awareness, binocular rivalry, and motion aftereffects. And as such, he is widely known as one of the most extraordinary neuroscientists in the area of visual attention. As such, it is my great pleasure to be introducing him to you today, and I would like to welcome Professor Sheng to the stage. Thank you.
First, I'd like to thank the organizers of this conference, Bruno and also Olivia, I'm not sure if she's here, for inviting me to share some of my work with you. And this is the first time I came to Australia and also of course, to Brisbane. And I know it's the winter time here, but for someone from Minnesota, it's practically summer. Now, in the title, you see that I also put up my name in Chinese. And the reason I did that is, as you can see, I also translated the meaning of my Chinese name. The family name, the last name, means the WH word. You can pick your favorite, okay? And the given name means life. So ever since I was born, I've been asking the most important question in life. But my father probably didn't know, but if he would know, he'd probably have given me a different name. And instead of asking what is life, maybe what is consciousness. Okay, I also have a joint position now at the State Key Laboratory of Brain and Cognitive Science in Beijing. So next time if you're in Beijing, feel free to drop me a mail and visit our labs. Okay, on to the presentation of the scientific content. I will be talking about primarily binocular rivalry and using binocular rivalry as a tool to study various aspects of visual awareness and attention. And there's a relatively long list of experiments and topics that I want to cover, so I want to probably go straight into the contents. Before I discuss specific experiments, for maybe a few minority in the audience, I'll briefly introduce binocular rivalry. Probably 90% of people in the audience know what binocular rivalry is, but just in case, this is when you have two different images presented to the two eyes, and the two images are different. You cannot reconcile between them, and you get into the alternating percept as sort of demonstrated here. That if you wear anaglyphic glasses, looking at the red and blue images on the left, what you will experience is something like this, alternating percept of the left and the right eye's image. Or another demonstration on the left here, you'll see the alternating images from the left and right eyes. And later I will talk about using a modified paradigm, it's called continuous flash suppression, to render images invisible. The reason for that is during binocular rivalry, although at a given time one of the two images is invisible, the dynamics is not really under our control, so it's not a convenient way to make a specific image invisible. Whereas with the continuous flash suppression, you'll have a lot more control to make sure that an image stays invisible for a fairly long period of time. And you can also make the images more interesting, and that it's still suppressed. What you perceive is still the flashing noise. And for people in the audience who still have trouble understanding binocular rivalry and continuous flash suppression, here's a more real-life explanation. That's binocular rivalry. Well, not binocular, that's rivalry. And that's continuous flash suppression. Apologize to fans of the Brazilian team. I know probably there are more Germans here, so I'm safe. Especially with Robert here, will come to my defense. Okay. Now, onto the experiments. The first set of experiments I want to describe is related to binocular rivalry per se, not related to awareness as yet. But I think it is important to have a better understanding of the mechanisms of binocular rivalry before we can feel comfortable using it as a tool to study other things. Right? So with binocular rivalry, many people have examined the mechanisms associated with rivalry. And the questions that have been asked for many years is whether binocular rivalry, in Randolph-Blake's term, transpires in the earlier stages of the visual hierarchy or in the later stages of the hierarchy. Right? So that has been the question that's been addressed for many years. Of course, you don't really need me to remind you that the visual system is massively parallel. It's not just a single lane highway going from one point to the next. It has many parallel projections. And among them, the two major parallel pathways, of course, is the magnocellular and the parvocellular pathway. And with that in mind, we can ask the question whether binocular rivalry occurs or is supported equally in these parallel pathways. Right? That's a fair question to ask. And indeed, there are very clear hints from earlier studies, for example, from Robert, that shows when you have two stimuli that are projected to the two eyes that only differ in their temporal properties, like two different temporal frequencies, but without any form difference, you do not experience binocular rivalry. So the flicker frequencies are different when they're presented to the two eyes, and you see a combined version of the inputs. And in one experiment performed by a former graduate student in my lab. Now he's actually in Australia, Macquarie University now, Macquarie University now. We presented two different stimuli to the two eyes, and they're very fast flickers, and they also differ in their color and shape. So, for example, one eye sees this, the other eye sees that. If you don't have form and color difference, when you only present two flickering stimuli like this, very fast flickering stimuli, and the same shape, the temporal frequency difference is small. In this case, we will experience an interocular or binocular beat, and this is sort of depicted here. If the difference between these two frequencies is 2 hertz, the modulation frequencies, the envelope modulation will be 2 hertz, and our observers can clearly experience that 2 hertz modulation. And of course, when you experience that 2 hertz modulation, that means the two signals from two eyes are combined. If you only see one eye signal, you should not experience the interaction between them. And whereas in binocular rivalry, one input suppresses the other. So in terms of conscious perception, you see one or the other. So there's this competing predictions. It's possible that you'll experience binocular rivalry, so you see one color and shape, and then alternating with another color and shape. And when you do see the red color shape, the temporal frequency associated with the stimulus that you're seeing should be the frequency of that red stimulus. And when you switch to the green one, you should see the temporal frequency of the green stimulus. Or you could see the mixed or the integrated version of the input, and that in that case, you should see the 2 hertz beat. But when we presented the stimuli to our observers, they actually experienced interesting combination of rivalry and fusion in that the color and shape are alternating, and they're engaged in binocular rivalry. You see the red exclusively for a couple of seconds, and then you see the green one exclusively for a couple of seconds. However, when you only see one color and shape, the temporal frequency that you experience is actually a mixed or the integrated version of the two frequencies. So in other words, a single stimulus, it's analyzed or processed in the parallel channels, and the information of features that are processed in the Parvo channels, like the color and the shape, tends to compete if there are conflicting information between the two eyes. Whereas the information, presumably the very high temporal frequency modulation, is processed in the Magno channel. In that case, the Magno channel, even though the two stimuli are different, they are integrated, the aspects that are processed in the Magno channels. And we also had a relatively direct manipulation of the stimulus to push it into sort of the Parvo versus Magno channel. This is not absolute, of course. It's sort of just biasing it to the Magno versus Parvo channels. And as you can see that when we made the two rotating radial grating rotate very fast, pushing it to the Magno channel, we tend to see very little alternation in the direction of motion, whereas when they're moving slowly, in that case, we can register the difference between for example, the phase between the two gratings. Then we see more competition between the eyes. We see alternating in the motion direction. It rotates clockwise and then switches to counterclockwise. Whereas in the high speed, you primarily just perceive the counterphase modulation. We also made another arrangement to the stimulus to sort of remove local form conflict. In this case, we presented a sine wave grating behind little grids that look like this. So in each unit, each little square, there's only a mean luminance modulation, so it goes up and down. But because of the sequence of this luminance modulation, globally, one eye's grading is moving to the right, the other one's moving to the left. But the motion is a global motion, and locally, each little square is simply just modulated in time. The luminance of that square is modulated in time. And even though when you cover one eye or the other, you see very clearly the motion direction to the left or to the right, when you have these two presented dichoptically, again, our observers do not experience binocular rivalry when the local foam conflict is removed. So that's the summary for the first part of my talk, that binocular rivalry seems to be primarily a property of the Parvo pathway rather than the Magno pathway. If you have conflicting information between the two eyes that are processed in the Magno pathway, they tend to be fused. Okay. There's some implications of this, I guess, proposal. I'll mention that briefly when we talk about the experiments next. So Sheng made the remark that maybe I'll talk about the processing capacity in the absence of awareness. I'll speak about the experiments, but maybe in the summary I'll discuss some of the implications. And we have done, as many other people have done, both behavioral experiments and neuroimaging experiments to show that in the absence of awareness, you've seen that yesterday and you've seen many publications reporting processing of many different properties, or unconsciously, some people use that term, and in the absence of awareness, it's maybe a little bit more specific. And again, as I highlighted here, they're both neuroimaging experiments and also behavioral experiments. Now, in terms of the tools that we use to make the, well, whatever information you presented not accessible by consciousness or in the absence of awareness, just as a reminder, this is the kind of manipulation, the continuous flash suppression that we use. And we can use different images, present different images, and still remain suppressed. So many years ago, well, now it's many years ago, another former graduate student, now he's in Beijing University, Fang, and I did a fMRI experiment. And this is, of course, related to many discoveries and theories from Mel Goodell that show the dissociation between action and perception. But instead of doing or making the observations in patients, in this study, we, in a sense, demonstrated that in normal observers, when you made the images invisible, in this case, using the continuous flash suppression, back then we didn't know the name. Tsuchi didn't tell us the name, so we thought we were using a modified binocular rivalry, so make one stimulus, the noise stimulus, much stronger than the intact images. So we call that modified binocular rivalry, but essentially it's a CFS technique. And now, when you present the face or a tool, you can present that either visibly without the suppression noise, or you can present the face or the tools invisibly with the interocular suppression noise. So we have the visible condition showing in the gray bars. These are individual subjects' activation. The top panel showing activation from the dorsal pathway, the bottom panels showing activation from the ventral pathway, and the gray bars showing individuals' activation for the visible condition, and the dark or black bars showing activation from the invisible condition or the suppressed condition. The only thing I want you to notice is the unique results here, that when the images presented were tools and they were presented invisibly, the black bars, from the dorsal pathway, the FMI measured response was still very robust compared to all the other conditions. When you present a face visibly or invisibly, you see from visible to invisible, dorsal and ventral, the activity goes down to close to zero, whether it's dorsal or ventral. When you present the tools visibly, you get strong response, but when the tools were rendered invisible, the activities in the ventral pathway goes down to close to zero, but in the dorsal pathway, it still is very robust. So that shows a dissociation between the ventral and dorsal pathway under the sort of conscious and unconscious conditions. And I don't really need to sort of describe the potential interpretation of this result, that the dorsal pathway activities are relatively more independent of conscious awareness, and because it's related to action. And when you use action-related inputs, then you can reveal the property of this dorsal pathway. Another neuroimaging experiment that we performed also quite a few years ago now with another former graduate student, now also in Beijing, Yi Jiang, used, again, continuous flash suppression, and now with faces either of the neutral expression or faces with fearful expression or scrambled faces. And just briefly, what we observed was, well, the motivation of doing that study is to see whether any face-sensitive regions in the brain are still capable of processing face-related information in absence of awareness. And as you can see that with the two key areas identified, the supratemporal sulcus area and the fusiform face area, the activities in these two areas, of course, are very robust. When faces were presented visibly. And I want to just point to the change from the red curve, well, no, not from the red curve, from the visible condition to the invisible condition in the fusiform face area. Should also note that the scales are different. The activities from the visible condition compared to the invisible condition, the visibility effect is huge in the FFA. So it's about 1/5 of the response level when the images were presented invisibly. But the reduction is relatively uniform, whether the faces were of the fearful expression, that's the red curve, or when the faces were of the neutral expression. In both cases, you get a slightly higher response level to the fearful expression in the FFA. But in the STS, what you see is that going from the visible condition to the invisible condition, the responses is essentially dropped to zero with the neutral faces, but remain fairly robust for the fearful faces. So this is about half of the visible conditions response level, but when the expression is of the neutral expression, the response is practically gone. And you get similar results from the amygdala, and we also looked at the correlation of activities between FFA, STS, and amygdala, and also as a control between V1 and amygdala. What we observed was a high degree of correlation between activities in the STS and amygdala, not between FFA and amygdala, and not between V1 and amygdala. So it looks like the activities in amygdala and the activities in the STS are related. We don't really know which one feeds to which, because fMRI signals are very slow, but in both the STS and amygdala, they're sensitive to the fearful expression of the face, even when the images were rendered invisible. And just quickly, we also examined the responses to the gaze direction on the face. When the faces were rendered visible or invisible with similar manipulations. And as you can see that the gaze direction could be a direct gaze direction, so from the observer's point of view, the face on the screen is, or the person depicted on the screen is looking at me, at the observer, in the direct gaze, or in averted gaze, the person is looking to the side. And in the visible condition, you see, I'll just highlight the results. This is a recently performed experiment. I just highlight the results from the right STS. As you can see that in the visible condition, when the face is clearly visible, you get higher activity to the direct gaze. So the person picture depicted is looking at you compared to the averted gaze. However, in the invisible condition, we get actually the reverse. And we have some idea of why this is the case. Don't really have time to get into the speculation, but I want to just show you this result to show that in the invisible condition, when awareness is removed, it's not simply just a weaker version. Whatever you see in terms of the brain's response is not simply just a weaker version of the conscious processing. So actually that's a summary of this part of my talk. A lot of the information can be processed in the absence of awareness and with sort of a vague term, especially the information important for our survival. So this is, I already made the second point that unconscious information processing is not simply a weaker version of conscious information processing. But related to the discussion from yesterday, after Zhiteng's talk, there are questions about what kind of information, a combination of features could be processed. And you probably can imagine that this kind of high-level information, like facial expression, presumably involves conjunctions of features, or the ability to process the conjunction of features. This is different from using relatively simple shapes and colors and forms to study what kind of information can be processed without consciousness in the absence of awareness. And I think the difference probably lie in different pathways. So with simple geometrical forms and the combination of forms and combination of form and other types of features, motion or color, you're probably addressing the typical processing pathway and presumably very heavily relying on the genetically destroyed pathway that we normally rely on for sort of conscious vision. Now, with what I vaguely termed important information, in this case, facial expression, and I'll describe other categories of, again, in vague terms, important information. We probably not exclusively or not even primarily relying on the genetically destroyed pathway. Probably are relying on, as again, I refer to Mel Goodell's previous proposal, that the subcortical pathway probably is more involved. And this could be a potential difference between the simple geometrical forms and other typical features that we encounter in vision research versus this relatively high-level information, but information that is important for our survival. Maybe I'll mention this, bring back this point again when we look at other relevant experiments. So that's a summary of the neuroimaging experiment. We also have performed behavioral experiment or psychophysical experiment to examine similar questions. And in one study that we used, again with Yijiang, used the, binocular rivalry as a tool, in this case also sort of similar to continuous flash suppression. Instead of keeping the information continuously suppressed, we purposely made the to-be suppressed image, in this case, in this example, upright face, not constantly low in contrast. If we want to keep it suppressed for long periods of time, then we will make it stay low in contrast, for example. But in this case, we gradually ramped up the contrast of the face and used a different measure, in this case, a very simple measure, simply just when does it take for this face, this intact image, to break from interocular suppression. And that time, that response time, is used as the dependent variable, as a measure of the strength or the level of processing of this image when it was suppressed, when it was invisible. And the key here is we need to emphasize to our subjects, our observers, that they do not need to know what this image is when they make their response, when they push the button to say, I see it on the left or I see something on the right. They only need to distinguish something that's coming out from the noise on the left or on the right. Just push a button, say it's left or right, something's coming out. I don't need to know what it is, okay? And with that, we are hoping that this sort of keeps the processing relatively restricted to the unaware stage. Well, I don't need to show you this again, but this is just for people who not actually have done this kind of research. This is just to give you a, right. This is the kind of experience that you get, left or right. It's on the left, right? You see something there. Of course, this is not the real experiment because you're not viewing it from a stereoscope. But this is the upright face, right? So if you only can see it now, then you have some trouble. So that's the upright face. You can just also experience what happens with inverted face. Right? You see that on the right. Okay, so that's the kind of experience that our observers have when they're performing this experiment. And what happens is, when you do it like this, our observers will press the button faster when the images presented were upright faces compared to inverted faces. But when you blend the face image to the noise directly, not presenting them interocularly, one to the left, one to the right eye, you present them on top of each other, just very gradually ramp up the contrast of the face, and we call that the control condition. You don't see the difference between the upright and the inverted face. And this sort of suggests to us that during the suppressed duration, when the upright face was suppressed, all the inverted face was suppressed, something is different in terms of processing of these two images. And that difference made the upright face, which is depicted, the black bar, the response time, is significantly shorter than the inverted face. And you can maybe take it a step further to say that the brain, somewhere in the brain, the information about the upright face versus the inverted face, or that difference is registered. And that difference cannot be very low level difference, right? Because it's simply just the inversion of the face. And other people have used this paradigm to study the processing of faces, but now adding the expression information to the mix. And I don't need to explain the details of this, but with this kind of paradigm, with this particular measure, they also observed first the inversion effect. So this is upright, this is inverted. Upright tend to be faster than the inverted, but also an effect related to expression. But how do you explain that? I don't know. Fearful face come out faster than neutral face, but happy face come out of the suppression slower than the neutral phase. Maybe the subconscious system is happy to stay with the happy phase without getting into the conscious system. And there are other studies. I'm only listing a very few here. And with this paradigm, there are probably in the maybe around 20 studies now have done using this paradigm. And I mentioned the gaze experiment in our lab just a moment ago with neuroimaging technique, but we also can use the suppression time as Timo did, Stein earlier, but we simply just want to see that the dissociation between the visible phase and the invisible phase with regard to the gaze direction. In the visible condition, we get higher response to the direct gaze compared to the averted gaze. But with the invisible condition in our neuroimaging experiment, we actually get higher response to the averted gaze than the direct gaze. We want to see whether the condition that we used is different from others. And in the invisible conditions, something is strange about the averted gaze. But when we did the behavioral experiment using the suppression time as a measure, we got results consistent with previous studies showing that the direct gaze also break from suppression or emerge from suppression faster than averted gaze. And that shows even for the same condition, same stimuli, the neuroimaging results and the behavioral results are also telling us different things. We can't really just take the activity level, whether it's higher or lower, as a direct measure of sensitivity. Okay, so this is presumably showing we're somewhat more sensitive to the direct gaze as manifested in this behavioral response. But in the neuroimaging experiments, in the invisible condition, the cortical response to the averted gaze was actually higher than the response to the direct gaze. We have also tried with biological motion stimuli with this paradigm, the suppression time paradigm. The idea is to see that when you removed awareness, when you don't have direct awareness of the stimuli, when we initially did this experiment, we were interested in seeing whether the unconscious brain is capable of linking spatially disconnected information. So in order to see the worker, you have to integrate information from the individual dots. They are not connected, right? So that's the original intention for doing this experiment. And of course, with the intact point light worker, you can clearly see a person walking to the left or to the right. And even for the inverted version, you can also see the Spider-Man walking upside down on the ceiling. And the scrambled version maintained the trajectory of each dot, but the spatial position is destroyed. So here you have just basically all the dots moving just as this one, but their location is scrambled. Here you also have all the dots moving just as this one, but their spatial location is also scrambled. So we included these two scrambled conditions as a control condition, right? So as a baseline condition. Our intention was to see whether this upright worker and inverted worker, just like the phase experiment, the upright phase come out of suppression faster than the inverted phase. With the biological motion stimulus, we want to see whether the upright worker come out of suppression faster than the inverted worker. And we were surprised by these results. In fact, when the point light worker was intact, whether it's upright or inverted, they came out of the suppression equally fast. There's no difference. And interestingly, the difference occurred or appeared in a scrambled condition, so between the two scrambled stimuli, this one came out of suppression faster than this one. Or I probably should say this one came out of the suppression slower than this one. The reason for that is it seems that whether it's upright or inverted, once you get the information, the first point I want to conclude is the unconscious system is capable, I think, of integrating the local information. But I want to emphasize this part of the result, that when you scrambled the location of the dots, the configuration information is gone, but the local motion trajectory is still intact. And in our brain, somewhere in our brain, we are still sensitive to that local biological motion information. And that tells us that this stimulus is unique, this stimulus is special. We need to somehow enhance the processing of this stimulus. And I think it makes sense that from an evolutionary perspective, that when we were in the woods, we probably don't want to have a system that waits for all the pieces integrate together before we decide to run away. So if we just get a glimpse of a tiger moving in some way, we probably should decide to, well, either pay attention or start running. So that's the conclusion of the behavioral experiment related to the sort of the processing capacity in absence of awareness. And the conclusion is essentially identical or the same as the conclusion I gave, or the summary I gave for the neuroimaging experiments. So information important for survival seems to be able to be processed in the absence of awareness. And also the behavioral experiments also suggest that in the processing of the information when awareness is removed, it's not simply a reduced version or a weak version of the conscious information processing. So these are the 2 points I want to highlight from both the neuroimaging experiment and also from the behavioral experiment. Now, I mentioned that the Magno and Parvo pathway study that I presented at the beginning is relevant for our interpretation of the results. I just use one slide to make that point. If you believe what I presented, that the Magno and Parvo pathway differentially engaged in binocular rivalry, then when you use binocular rivalry as a tool or continuous flash suppression as a tool, and again, this is a very simple depiction, schematic depiction. If this is the suppression noise and this is the stimulus that you want to see whether it can be processed, and let's say in the Magno pathway, you get this. In the Parvo pathway, you get this, the strong interocular suppression. Because this stimulus is much stronger, you end up with this one being dominant, the other one is suppressed. Whereas in the Magno pathway, let's say you get the integration of these two, and the integration of these two gives you this. And whatever the information that you have is not necessarily gone, but it's very, very, well, I don't know if it's a real word, and unsalient or very difficult to process. So it becomes a normal masking situation. So if your purpose is to see whether consciously suppressed information could be processed, then maybe you want to give the system a chance by avoiding this kind of situation. Okay. So the third part of my talk is related to rivalry and attention. So we have many studies published, and again, from people in the audience, and discussing, showing the relationship between awareness and attention or consciousness and attention, whether they're the same, whether they're different. I noted that another keynote address, I think tomorrow, will be focused on this topic, the relationship between consciousness and attention, and now also published extensively on this topic. But I will present a few experiments to see whether they can sort of convince you to believe in one way or the other. But I probably don't want to spend too much time talking about the relationship in abstract, but I'll just get into the experiments. The first experiment examined the issue of whether attention can be influenced by information that you're not aware of or you're not consciously aware of. Okay, maybe that's a redundant word. And the way we did that is to use Posner's, very established attentional cueing paradigm and the cue validity paradigm. You have fixation, the target could be presented on the right or on the left, and before the target was presented, you have a cue presented on the right or left, and you end up with the target on the cued side or the target on the uncued side, or the valid cue condition or the invalid cue condition. And of course, your performance will be more accurate and faster when the target is presented on the cued side compared to when the target is presented on the uncued side. That's the typical cue validity paradigm. And we simply just replaced the cue frame by interocularly suppressed, potentially attentional cue. We don't really know when something was suppressed could still serve as an attentional cue to attract your attention to that side. And this is presented to the dominant eye, this is presented to the other eye, and as you can see, the two sides in the suppressed eye contains very different information. Presumably one side is more attractive than the other for some people. But at the conscious level, what our observers experience is just two patches of noise. They're identical, there's no difference. And then after this frame, we presented a little probe, either on the side where the intact image was, or on the side where the scrambled image was. And the sides are, of course, counterbalanced. Now, with this potential cue, the question is whether performance will be better when this is presented on one side versus the other. And of course, we got the positive results, otherwise we wouldn't be presenting this. Now, initially, when we looked at the results, it was pretty confusing until we separated the results by the gender of the subjects and also the gender depicted in the images. And we took the images from the IAPS, International Affective Picture Set, and we initially included many different kinds of images, faces and flowers, this kind of thing, positive, negative images. But in the pilot experiment, we noted that the erotic pictures were the most effective ones in terms of directing people's attention. So we ended up doing most of our experiments with the nude pictures. And as you can see, the vertical axis showing the attentional effect. Positive means attracting attention, negative means repelling attention. And depending on the gender of the subjects and the gender depicted in the images, you get the opposite gender's picture attracting your attention, and the same gender nude picture somewhat repelling attention. And this effect actually was better characterized based on the sexual orientation of the subjects rather than the gender of the subject. In other words, if you are homosexual male, you behave more like heterosexual female, that your attention is attracted to a male nude picture. So that's the well, it's not dwelling on the sort of the gender or the sexual orientation effect. The basic point I want to make is something important, but it's invisible in your visual field. Somewhere in your visual field can attract your attention or push your attention somewhere else. So it has a very strong attentional effect. And of course, this has also been demonstrated before. The eye gaze information can also affect where you direct your attention, even when the gaze is invisible. This is when the gaze is invisible is on the left side, on the right side. When you presented a face with gaze either to the left or to the right, and then that can serve as an attentional cue, even when the face with the gaze is rendered invisible. So when the test probe is presented on the side where the gaze is directed to, you perform it fast. This is the reaction time, faster here. And when the probe is presented on the opposite side where the gaze is directed to, you perform a little bit slower on the probe task. And of course, with the biological motion stimulus, the scrambled biological motion stimulus in particular, where in one stimulus the local motion is maintained, in the other stimulus the local motion property is destroyed by inversion, we also get attentional effect. In other words, you can present two patches of seemingly randomly moving dots, but one side you preserved the local motion information, on the other side the local motion information is destroyed. You end up with the side where this is the scrambled condition, this is the performance as a measure, the side that has the upright scrambled stimulus, which preserves the local information, the local motion information. You get a difference between that and the inverted scrambled biological motion stimulus. In other words, even though you don't really know which one has biological motion, our naive subjects cannot tell the difference. Their attention was attracted to the side where the potentially biological motion information is present. Okay. Again, we have objective measure that our subjects, naive subjects, could not tell the difference between the two sides. This is sort of another way of sort of achieving unawareness. So this is unaware of a property rather than unaware of the stimulus. So this is the summary of this part of the talk, that information is processed can be processed in the absence of awareness. And that has been demonstrated, but this couple of experiments I just described tells us that this is not just an epiphenomenon. The information processed in the absence of awareness have real consequences. They can direct our attention to one place or the other. And also highlighting the point that, again, information processed in the unaware condition is not simply a reduced version. I see that my time has getting close to be used, so I'll just spend a few minutes summarizing the next couple of experiments. This is quickly just to show you that something, again, you don't have conscious access of, can guide attention. Now, attention modulation, we have seen that many times in behavioral experiments and also in neuroimaging experiments. This is from Ted Dio's lab, published quite a few years ago, showing in fMRI when you have your attention directed to different parts of the visual field where the stimulus is constant, you can almost literally see the attentional spotlight illuminating different part of V1. Okay, so wherever you're attending to the corresponding part in V1, the activity becomes higher. Okay, so that's just showing that with fMRI, you can see the, well, attentional modulation effect, and even you can see that, again, with fMRI in LGN. But fMRI is a tricky business. We don't really understand the source of the signal, so we actually trust behavioral experiments more than fMRI experiments. The question is whether top-down modulation, attentional modulation can be eye-specific.